Are we live? Are we live, Seema?
Yes, ma'am, we are live.
Okay. Okay, Professor Kakkar, now you can introduce the speaker.
We'll wait for 2 or 3 more minutes, or
Okay.
Yeah, you might want to. I mean, since you officially said it's 11 o'clock, may as well wait.
yeah.
Yeah. So it's my clock is showing 10:58, so
Yeah,
2 minutes.
Yeah. So Devang, IIT is working fully online, or I mean, the classes are.
Except the PhD students are here.
Right.
Classes are online.
Classes are online.
Yeah.
We thought we've been teaching online also here, and the main thing is not having any eye contact with the students. They ask questions, but not seeing them feels very weird.
Yeah, it feels weird. But it's better than nothing, I think.
Yeah.
How's Rama doing?
Rama's fine. She started to go back to ICTS for I mean, I think TIFR has announced that they're basically resuming operations, and so she's been going in a bit. I was coming in twice a week. I was teaching in a classroom with no people in it. I was teaching, on a blackboard.
Okay.
Real blackboard, because we have a nice big blackboard.
Yeah.
As you can see in my office also, I have a 16-foot blackboard.
Yeah.
Real shock. But the room was, they had camera and sound and all that. And,
that is important.
It was reasonably set up. Wasn't brilliant because you had to switch. The board is very long, so you had to switch the camera board 1, board 2, board 1, board 2. So if it's on board 2, they can't see what's on board 1. But it's not terrible.
Yeah, they can just go back a little and.
Right, it's all recorded, so they can go back. Yeah.
So we'll start.
Yeah, sure.
Good morning. Welcome to the first lecture in the INSA Fellows webinar series. I am Devang Kakkar, currently the Vice President, Science and Society of the Indian National Science Academy. And I will be chairing this session. It's my pleasure to introduce Professor Sriram Ramaswamy, someone who I have known for a long time. Today's speaker. Professor Ramaswamy is a distinguished physicist who's renowned internationally for his pioneering and path-breaking research contributions in the area of active matter, the subject of his lecture today. Active matter is composed of elements that propel themselves and interact with each other. For example, a flock of birds. Professor Ramaswamy was among the first to develop the laws for the behavior of such matter and has worked in the area for the last 2 decades. His other areas of interest include suspensions and liquid crystals. Just a little bit about his biography. Professor Ramaswamy did his PhD at the University of Chicago in the Department of Physics, and then his postdoc at the University of Pennsylvania, after which he returned to India and joined the Indian Institute of Science in 1986 as a research scientist, and he has remained in the department since then, except for a brief stint when he spent 4 years as the director, as the center director of TIFR Hyderabad. Professor Ramaswamy is currently a professor in the Department of Physics at the Indian Institute of Science. Professor Ramaswamy has received several awards and recognitions for his outstanding research achievements, and I'm going to just sample a few and not list all of them. He serves or has served on the editorial boards of a number of prestigious journals, including the Journal of Statistical Mechanics, Soft Matter, Proceedings of the Royal Society A, Advances in Physics, Physical Review E. He's also been recognized by a number of bodies. For example, he's a fellow of the Indian Academy of Sciences, a fellow of the American Physical Society, and a fellow of the Royal Society of UK. He has received many important awards, including the Shanti Swarup Bhatnagar Prize, the J.C. Bose Fellowship, and the Infosys Prize. And I'm very proud to say that he is a fellow of the Indian National Science Academy. I now invite Professor Ramaswamy to deliver his lecture.
First, thank you, Devang, for that very kind introduction. It's really nice to have a friend introduce you. Thank you, Chandrima and Insa, for this wonderful invitation. I will start to share my screen. Let's hope it works. Okay, hold on. Okay, so I hope people can see the talk slide. You can see now?
Yes.
We can see it.
Yeah, thanks. Okay, so thank you. Here goes my talk. That's where I am, and that I get support from a fellowship from CRB and from the Tata Trust. And I'll tell you what this thing called active matter are so excited about it. By something as this is an odd state of self-driven, of living matter. And I'd like to be able to write down equations that describe an object like this. This particular object, I had put the caption, seems to have got lost. This is a movie by Surajit Dhara of the University of Hyderabad, and it's millipedes swarming on the University of Hyderabad campus. I think the caption has got hidden, sorry about this. And so this, to my eyes, it's a liquid crystal made of living things, and I'd like to make a mechanical, dynamical theory of it. That's the idea. Okay, so my talk, that is just a sampler. By full disclosure, I have not managed to make a dynamical theory of that interesting system. My talk will be outlined as follows. I'll sort of motivate the subject and tell you about early results, which actually still in many ways control the field. Then I'll pick a few specific topics from our recent work. I will focus on our own work and tell you in a little bit of detail about them. For the most part, without equations, sometimes with equations. Three quite different topics, and then I'll summarize. Okay, so the motivation really is that living matter, after all, is a kind of matter. And it's different in many ways. Obviously, it's different because it can take decisions, it's different because it evolves, it's different because it reproduces. But one of the fundamental ways in which it's different is that unlike a chunk of metal on your table, living matter is in a driven state. In some sense, you can think of it for most of its life as being in a kind of stationary but driven state. It goes on taking up free energy and using it to do something. And unlike, let us say, a wire connected to batteries at its two ends, every component of living matter is driven. Whether you talk about what goes on inside one cell or whether you talk about a collection of animals on a much bigger scale, there's a kind of similarity, a dynamical similarity from the smaller scales to the bigger scales, because until you go to until you break up the individual components of living matter, basically living matter is made of active stuff. So active matter is a category invented by condensed matter physicists to colonize even living matter with their techniques, their ideas, their approaches. And this subject has really gone viral, to use a modern phrase. And there's a couple of references which I commend to you. One is a nice article by Rob Phillips on why if you're doing equilibrium physics, you're doing the physics of dead things. If you want to do living matter physics, it has to be out of equilibrium. And Leibniz, many years ago, spoke about living matter, if you like, as a machine made up of machines. And this is reiterated in a nice review article by Needleman and Dogic, Nature Reviews Materials 27.
Sriram, your slides are not being shared.
Oh, really? Oh my God. So you can't see the slide at all?
No. So can you stop sharing and start sharing again?
Okay, will do. It's refusing to stop sharing also. Okay. What's going on? Share not available, maybe 4.
Seema?
Yes.
Seema, can you check, Dr. Seema Mondal, can you check from our side if there is a problem with sharing?
Okay, ma'am. Ravi?
It's not even, so right now, what can you see on your screen from me? You can see me?
We can see you.
Yeah.
I see.
Just log off and log back on.
I think I'll just have to disconnect because it's not even allowing me to not share. Okay, I'll disconnect and reconnect. Okay, sorry about this, folks. Oh, wait. Now the camera has to change. Just a moment. Okay, so now let me share screen. Now tell me, what can you see?
We can see the screen now. Yes.
Okay, let me and
Full
now full screen also.
We can see.
Chalo, shukar hai. Okay, great. So my talk, as I said, I'll try to tell you why active matter has become such an intense area of research. You can still see my screen. New slide.
Yes, we can see your screen. Yes.
Okay, great. Okay, so let's take some. It's always good to start with some examples. So basically, any body of living stuff we refer to as active matter, but there are different scales on which you can see it. You can take stuff from inside one cell, like molecular motors and biofilaments and the fuel that needed to run the motors, and make artificial active matter. You can look at tissue in 2 dimensions. This is an epithelium from experiments by Duclos et al. You can look at groups of birds flying, like this movie taken by my student Rahul Chajwa when he was in Hyderabad. Or you can look at artificial realizations. You can take a surface, a table, and you vibrate the table up and down, and you put on it an object with a shape which is such as to be able to convert the up and down motion that is out of the screen in this picture into motion in the plane. So you can create an artificial self-propelled object by the very seemingly silly expedient of just vibrating a surface and putting a pointy object on it. I'll come back to this problem. You can also create, this is a mechanical artificial active matter system. You can create chemical systems which are active by taking the particle partly covered with catalyst and putting it in a medium containing a compound that the catalyst breaks down. And that object will start running around and swimming because of a maintained gradient of reactant along its surface, which gives rise to osmotic flow along its surface. Or you can create electrical active matter. You can take a 2D material, let us say a 2D electron gas, and convert it, connect its two faces to leads at different chemical potential and drive energy through the face of this material. The physics in the two-dimensional plane of this material is then going to be some kind of matter physics with equations, of course, that one has to invent. And I won't say anything about many of these topics, but I will talk about some of them during my talk. Okay, and the aim basically is, there's a big challenge. What are the emergent laws governing living or other self-driven matter? And clearly, mechanics in biology is very important. Functionalities of living systems and how much you can imitate them is very important. And these are all things that I'm trying to do in this field. So active matter, therefore, consists of the components of living systems or maybe artificial realizations. At the level of the constituents itself, detail balance, that is to say, the balance between forward and backward processes is broken by virtue of the fact that you're constantly providing fuel to these systems. So these systems, at the level of the individual components, steadily convert free energy into some kind of organized movements. And collections of these objects are called active matter. And we have a sort of formal take on the subject in a couple of papers in the Journal of Statistical Mechanics a few years ago, which those of you who like technicalities are encouraged to read. Now, because we are condensed matter physicists, we like talking about ordered states. We are interested in symmetry and the breaking of symmetry. So it's important to look at the kind of symmetries that one can get in the resulting ordered states and the kind of symmetries that are available at the level of the particles. So you could imagine just a collection of droplets on which some chemistry is happening, they're running around. That, if you like, you can call scalar active matter, in the sense that there's no particular directionality except transiently on the scale of the drop. You can also imagine a particle which has a shape and a head and a tail and moves in some direction. Systems of that sort, which have a head-tail difference, we call polar. You can imagine objects like that epithelium, that two-dimensional cell layer I showed you, in which the objects are elongated, but there's no difference between head and tail. Those we call apolar. You can imagine things in between where the structure has no polarity, but the movement has polarity. And these kinds of objects can organize themselves into different states. Of course, they can be distributed isotropically. They can align with not only their axes aligned, but head forward and tail back. Or they, even if they have individual head-tail structure, they can align with only their axes, and so forth. So you can have a variety of different phases. Just like in equilibrium condensed matter physics, you can have different kinds of crystalline and liquid crystalline phases. And those are the kinds of things we are interested in. One place where you can read in great technical and conceptual detail about this field is a review article we wrote a few years ago, but it's still fairly current, which gives a very comprehensive view with lots of detail filled in. The dynamics of active systems, as with the dynamics of any material system, can take place in different regimes. You can imagine active systems on a dry solid surface, like this artificial flock in the work with Nitin Kumar, Harsh Soni and Ajay Sood some years ago, or you can imagine active systems moving through a fluid. There's clearly going to be dynamical differences because these guys, when they move, disturb the surrounding medium, which disturbs other particles. That will turn out to be very important. Now, one thing I should say is that the idea of studying active systems, and even the word active stress, we thought we invented it, but it turned out people had worried about this. Very distinguished chemical engineers had worried about it 50 plus years ago. In an article that unjustly is not very widely known. These guys, Finlayson and Sriram, talked about motion without the intervention of outside forces. They said that the necessary machines, necessary engines, operate in living systems. All of these are quotes. They said that this is virgin territory for research in complex fluids. They said a microscopic understanding will require a statistical mechanics not yet detectable on the horizon. They talked about making polymeric and colloidal systems whose properties would approximate living protoplasm. They talked about different kinds of active matter. In particular, they talked about one-dimensional contractile force as happens in muscles and in... and they also talked about systems in which you can get stresses of the kind operating in living systems by gradients of various scalar quantities. despite all this and despite a very detailed paper on the hydrodynamics of these systems, which incidentally, for some reason, didn't discuss the topics that came up many years later in our work, this paper did not launch a new field. It almost sank without a trace. And there's an interesting question in the history of science as to why it didn't happen. And I think the reason is that it was a little too early, and the necessary machinery for writing down hydrodynamic descriptions of the dynamics of phase transitions, of the dynamics of materials other than simple liquids like liquid crystals and superfluids, all that machinery was not yet available. The so-called framework of broken symmetry hydrodynamics and kinetics of phase transitions came along a few years later, so this paper somehow appeared before its time. So I discussed this in a sort of short contemplative piece in a review journal called Nature Reviews Physics, in an issue to commemorate 200 years of George Gabriel Stokes of Stokes's law and so forth, which some of you may find interesting. Okay, I'm moving at a rather leisurely pace. Let me now, I'm still in the introduction. So one way to study active systems is a rule-based or agent-based approach. You just say there are these things, they like to not bump into each other, they like to come close to each other without bumping into each other, and they like to line up so that they point in the same way as their neighbors. If you take a system like this and study it on the computer, you have things that look at their neighbors, line up with them, and move the way they're pointing, then as long as the error they make in lining up is not too large, and as long as the local neighborhood is heavily populated, you get spontaneously a state in which everybody moves in the same direction. Which direction depends on some accident of initial conditions. If the noise is too large and or the density is too small, you get an isotropic phase. And as a function of these two control parameters, you have a non-equilibrium phase transition, which is called the flocking transition. This is the Vicek model, which was invented not by Vicek, but by a man named Reynolds. And it's been widely studied. And this is the simplest kind of picture of active systems. And you can see this kind of behavior even if you take a collection of real motile cells, such as the keratocytes in these experiments, and you get a flocking transition in these systems too. Many interesting things are known about this system. The rule-based theory of Vicek et al. Was turned into a continuum field theory by Toner and Tu. It was shown that these systems could actually exhibit true long-range order even in 2 dimensions, contrary to the prejudice that theoretical physicists might have from analyzing magnetic systems in 2 dimensions. But flocks in fluid, and this is where we came in, flocks in fluid are different because when they line up, the act of aligning, the act of moving, disturbs the fluid, which disturbs other particles in the system, and a lot of fun things happen. So what we did was, our approach really was not to take a given class of experiments and try to do theory to model that experiment. Our approach was to ask a general question. If you've got a collection of objects that are motile and trying to line up with each other and in a fluid, how is this different from the physics of a collection of dead particles which line up with each other in a fluid? The answer, the equations of liquid crystal hydrodynamics, we formulated about 20 years ago at this point. And our approach was vindicated in the sense, basically what I'm talking about is looking at systems like this, collections of these objects. Here, of course, it's on a surface, but collections of motile things and what they do to each other in presence of a fluid. And our general theory, general symmetry-based approach was vindicated when a couple of years after us, groups at Paris and Dresden formulated essentially the same equations in an attempt to describe something on a much smaller scale. We were thinking of motile organisms in a fluid. They were thinking of the world inside one cell, where you have filaments and motors and chemical fuel, and the molecular motors and filaments together make that system into a fluid of orientable active entities. So there's a lesson here already, which is if you think about the problem based on symmetry and conservation principles alone, you will make a theory whose regime of applicability is much larger than the area for which you designed it in the first place. So what we did was to write down equations for an orientation of a bunch of objects and the flow field of the objects plus the ambient fluid. And we said the orientation responds to the flow or the orientation tries to relax to equilibrium in the absence of flow. The flow is driven by these filaments, in part because they're trying to line up with each other, which would happen even at thermal equilibrium, but in part because each of these objects, each of these orientable objects, because it is self-propelled, carries a stress, a local anisotropic stress, just dependent on its orientation. And this coupled dynamics, orientation responds to flow, orientation produces flow, gives you all kinds of dramatic it says that if you've got a bunch of orientable objects in a fluid, then if you line them up, then depending on whether they are this kind of swimmer, which swims by pushing fluid backward and moving forward, or this kind of swimmer, which swims by pulling fluid towards itself and moving forward, you will produce a system in which, for these guys, the suspension is less viscous when they're alive than when they're dead. And for these guys, suspension is more viscous when they're alive than when they're dead. This striking qualitative prediction which we made 18 years ago has been the subject of experimental studies over many years. In fact, the most recent is probably even more recent than 2015. And it beautifully confirms what we predicted on the general terms. We also argue from the same equations that if you have an aligned state of these guys, you can't actually make a bulk aligned state in a dynamics where viscosity dominates. If you've got a bunch of aligned filaments, they're swimming. If you disturb the orientation slightly, then the disturbed state will give rise to flows, which you can see here. These filaments closer together, those filaments farther apart. Therefore, the force is concentrated here than here. As a result, you get a flow this way here, flow this way here, and that topples the filament, that drives the filaments more in the direction in which you perturb them. If they were swimmers of this type, they get distorted. Their order gets destroyed not through splay but through bend. So basically, this suggests that an aligned state of active or self-propelled particles in a bulk fluid is simply impossible. It dissolves into chaos no matter what you do. We'll return to this point. A detailed confirmation of these predictions in specifically pursuing our instability really came only 2019, slightly over a year ago, in experiments on an active liquid crystal made from microtubules extracted from living cells, molecular motors extracted from living cells, and their fuel. And they placed them in various kinds of geometries, and they exhibited exactly the instability that we predicted. There was other fun evidence for such effects in the fact that if you take a single cell and put it on a fixed bound geometry, and you watch not the cell but the nucleus inside the cell, which is what I show here, then flows set in the surrounding medium due to a similar instability, which then makes the nucleus rotate. So this subject is still a very live subject, and we've been studying it. I would recommend that you look at our papers over the past couple of years, which I will not talk about what happens when you confine active fluids, what happens when the system is not a fluid, but an active solid, and so forth. Many of them are with my former student, Ananya Maitro, who is now a postdoc in Paris. Continuing with early results, you can also think about systems in which the order is not that of things with a head and tail, but just a bunch of rods which align their axes. Imagine a bunch of rods on a solid surface, but imagine that those rods are active in the sense that maybe they have more kinetic energy for moving along their length than transverse to their length. Is an aligned state of those systems the same as a dead liquid crystal, or is there something different? We predicted, again many years ago, there's a big difference, that even though the aligned state on average isn't going anywhere, if you perturb such a system, either by splaying their orientation or by bending the orientation, that creates a local vectorial asymmetry. Once you have a local directionality specified, then in an out of equilibrium system, you'll get movement in that direction. That movement, it turns out, gives rise to enormous fluctuations in the number density, which were confirmed in experiments by my then student Vijay Narayan and Narayan Menon and me some years ago. And it also means that if you have defects in the structure, of which there are two kinds, you can have defects of this type or defects of this type. Here, what we predicted was defects of this type have a polarity, so they should move, and Vijay's experiments showed exactly that. If you so even though the individual rods are head-tail symmetric and don't go anywhere, the structure has a polarity, and that polarity of the structure gives rise to directed motion. This turns out to be hugely important to the physics of active pneumatics, and we predicted this on general symmetry grounds back in 2007. We were so casual that we predicted in the supplementary material of the paper, but we did make it very clear that the curvature around these defects gives rise to directed motion. This idea has turned out to be kind of dominate the subject of active pneumatics in recent years. So the reason why active matter has gone viral is sort of reasons from biology, reasons from physics. From physics, it's a new frontier for non-equilibrium stress mechanics and possibly a new frontier for material science. New kinds of order, weird behavioral defects, a strange kind of turbulence, which I'll talk about. It turns out, even though the particles have no particular directionality, they can condense into they can have something like a vapor-liquid transition without intrinsic attraction. There are implications for information processing. In the biological context, as I said, our active particle hydrodynamics turned out to be the equations required to describe the dynamics of the polymer skeleton of living cells. These ideas, not from our work, but other ideas on active phase separation, turned out to be the physics behind liquid-liquid phase separation in cells. Hydrodynamic approaches and topological defects have turned out to be important to describe the physics of the development, that is physics of development biology. And we got from work, especially on bird flocks in Rome, all kinds of insights into how best to describe them in physical terms. There's also the beginnings of being able to imitate some of the functionalities of living matter, which I will not have time to do. Okay, so now half an hour into my talk, I will start telling you about our current work. You remember I told you about flocking models, and you remember I then said that when you put flocks in fluid, the physics is very different. I told you that aligned states of swimmers are intrinsically unstable. But there was a catch. This was in a regime where whatever disturbances you put into the orientation produce a response in the velocity field everywhere instantly. That is, we didn't take into account that systems with inertia have a lag between when you force them and when they respond. So if you take into account the inertial lag and you take into account that these things are not just filaments, but filaments with a head and tail being different, how does the story change? So this is work we studied with my student Ryan Chatterjee, who is now at the University of Pittsburgh, and collaborators including the late Aditi Simha, Navdeep Rana, and Prasad Devlikar at the IIT Hyderabad, which you can read about in this preprint. Basically what we showed was there's an important control parameter telling you about the competition between how fast things are swimming and how fast the instability is setting in. When you take inertia into account, you get two speeds, the swimming speed and the speed defined by the ratio of the intrinsic stress to the local mass density. This quantity, in the original analysis in our work in 2002, we were effectively at this quantity equal to 0. And the question is, what happens when it's not 0? What happens when you take into account the inertial lag? So it turns out that when I will not inflict the equations on you, it turns out when you take those same equations, the growth rate of the unstable disturbances in the theory we initially wrote down as a function of wave number, that is the inverse of wavelength, would approach a non-zero value when you go to the largest length scales. The moment you include the inertial lag, it turns out it matters how much that lag is. If inertial effects are small, this picture holds almost out to the largest length scales. The more important you make inertial effects, the faster you make the self-propelling speed, the less important this instability is. It goes from being an instability at all scales to being an instability that grows linearly with wave number, to being an instability that grows as wave number squared, to going away altogether. So fast enough swimmers can escape this instability. That's what we showed. And a full phase diagram, if you like, as a function of one parameter which tells you the relative rate of orientational relaxation and velocity gradient spreading, that's over here, and our ratio of these, the squared ratio of the two speeds, says that if inertia is very unimportant, you get a state of weird, messed up defect turbulence. The more important you make inertia by, for example, increasing the speed of self-propulsion, the more you can escape this instability. You enter a regime where you perturb the initial state and disturbances seem to grow but slowly, but in fact you have an ordered state. So what you have is, when you're in the matter, you get extremely strong defect turbulence, which you can see over here. There's actually three-dimensional things. You get hedgehog-like structures, and it just dissolves into isotropic chaos. If you're in the regime where the instability is weaker, it turns out that even though an initially aligned state starts to have disturbances that grow, the disturbances do not destroy the order. The system stays ordered but noisy. So as a function of self-propulsion speed, if you like, you go from an utterly chaotic isotropic state to a noisy but ordered state. And then if self-propulsion is large enough, you don't even have noise, you just have a boring ordered state. But this is important. You go from isotropic defect chaos to noisy but ordered. So you actually have, as a function of self-propulsion speed, a phase transition from a state where orientation is lost and the order parameter is 0 to a state where you have order. And correlations grow as a function of, correlation is a function of distance. As you change parameters, change self-propulsion speed from small to moderate to large, the correlations get more and more slowly decaying. The correlation length associated with these correlations grows as you approach the transition. I've shown here the inverse correlation length as a function of the inverse of that control parameter, and that quantity shrinks. This means the correlation length grows. So that's one story which I wanted to tell. The inertia suppresses turbulence, inertia assists flocking. Okay. All right. Now let me go on to a different topic. I have, I guess, 30 odd minutes. Let me skip ahead through this and go to a rather different topic. I'll come back to the defects. Okay. Let me talk about something that I've been currently thinking a lot about, which is non-mutual interactions. You know that, in ordinary life, interactions are by no means mutual. Someone can be attracted to someone else, that person can be repelled by the first person. This idea actually operates even in the flocking of locust swarms, where it turns out, as was shown by Vishnu Guttal and others then in Ian Cousins' group in Princeton, that you can get locusts to align with their neighbors simply by the locust at the back pursuing the one in front because it wants to eat it, and the one in front trying to run away because it wants to escape. And that's clearly a non-mutual interaction. This one moves to catch that one, this one moves away from it. So, and the ideas of non-reciprocal or non-mutual or asymmetric interactions are very common in many settings in biology, including neural nets. We've studied this. We've produced a purely mechanical realization of non-reciprocal attraction, which I'll show you about. This the topic of non-reciprocal interactions is very current. My former student Surabhi Saha, now a postdoc with Ramin Palestinian, colleague Christina Marchetti and her student Vincenzo Vitelli's group, many people have been studying this. Basically, when you have interactions not governed by a pair potential, not governed by an energy function, really many, many new things can happen, and that's what this part of the talk is about. Okay, we actually worried about non-reciprocal interactions again many years ago, back when we were starting to think about self-propelled hydrodynamics. Jayjit Das, Madan Rao, and I were also thinking about generic non-equilibrium equations of motion for magnetic systems, seemingly a completely different topic. And there we said that supposing you have particles interacting on a lattice with a fore-aft asymmetry, then it could happen, if you're out of equilibrium, that the exchange coupling felt by the guy in front due to the guy behind can be different from the exchange coupling felt by the guy at the back due to the guy in front. If that happens, you get a very different kind of magnetic dynamics. This kind of dynamics, whose features I will not tell you about, except that you get all kinds of wild instabilities, cannot happen if the interaction is governed by an energy function. So it's an intrinsically out of equilibrium feature, which can only happen in the system is in some kind of driven state. We recently started thinking about this idea in the context of self-propelled particle systems. So I'll tell you a little bit about the story of pursuit and capture interactions in dead but activated matter. Done by my student Rahul Kumar Gupta, with Harsh Soni, my former student, Roshan Kant, a joint student with Ajay Sood and me, which you can read about in a paper which is on the archive currently. So let me tell you how that works. Now, some years ago with Nitin Kumar, we had studied collections of self-propelled particles in a background of these beads. And some of you may have heard talks on this from Ajay Sood. So you have a collection of these. This is, again, it's a top view of a system which is vibrating out of, that is, you've got a surface and it's vibrating vertically. You are looking at it from the top. That vibration is turned into self-propelled motion by these macroscopic 5 mm long rods placed on it. And there's a background of little spherical beads. And nothing much seems to be happening. If you take the same system, keep the same number of those rod-like particles, and just increase the concentration of beads, now the way the rods move is totally different. The rods move, their movement moves the beads, and the bead movement aligns other rods in this medium. And you get a spontaneous ordering transition, which in this picture is that everybody starts going around clockwise. If instead you took a system in which there weren't boundaries, you would get a globally ordered state of everybody moving together in one direction. So this is a flocking transition. So our question is, this happened when the bead density was moderately high, but not very high. Supposing you make the bead layer even more densely packed so that it's a crystalline layer, what happens? Well, empirically what you see is, and this I think is from a direct mechanically faithful simulation, you see that when the rod moves in the in a fluid bead medium, it drags the beads with it. When the rod moves in a medium where the beads are packed to a crystal, it still moves, but it doesn't drag the beads. The beads are left behind. We just numerically colored some of the beads to show you this effect. If you look at the flow field around the rod, when the beads are in a fluid state, you can see nice, coherent, two-lobed flow field. When the beads are packed into a crystal, the rod still travels, but the beads move back and forth here and there a little bit randomly, no systematic flow. So what does this system, what does a collection of rods do in a crystalline medium? We'll answer that question for 2 rods. And so we actually made up a theory first. And we said that, look, let's look at what the system does. You see this rod moving, and it's clear that this rod is pushing locally on the bead medium. So this rod is like a local point force on this medium. So we said the rod moves, there should be a dot, this should be R dot, this should be the rate of change of position of the rod, moves in a direction determined by which way the rod is pointing at some fixed speed V0. The presence of a rod at position R produces a local force on the bead medium. The bead medium otherwise evolves so as the bead medium is described by a displacement field, which tells you the local pushing away from mean position of a local particle, and it tries to minimize some kind of elastic energy. And if a bead moves, it is damped by a substrate. So this is the equation of motion of the medium. If it weren't for the presence of the other rods, it would just try to relax to a state where it minimizes some kind of elastic energy. Because you've got a motile rod in it, that forces the medium locally. Moreover, another rod placed in this medium, if you impose a strain on the medium, that strain will reorient the rods. And how that happens is given by this equation. So those of you who like equations can inspect these. All that's being said here is rod follows its nose and moves. Rod forces the bead medium. Strains in the bead medium reorient the rod. Okay? It can reorient either because the crystal planes are curved. There is a local uniaxial strain. We will worry only about this part. Okay? A local strain in the bead medium reorients the rods. And the question is what happens? So you can solve these equations and you can work out the field, the displacement field. Basically, if you take this medium, how the sum is distorted on average due to the moving rod, you can actually calculate that analytically, and you find you can express it in terms of well-tabulated functions called Bessel functions. The crucial feature from here is that you sit in the frame of a moving rod and ask how the bead medium behaves. You find the bead medium, the distortions in the bead medium are highly screened in front of the moving rod and not very screened behind the moving rod. In fact, it turns out you have an exponential decay of distortions in front and only a 1 over square root of x distortion behind. This is a mathematical property associated with the functions in terms of which we are able to calculate the solution. We can check this. It's a little hard to check this in the real experiment because the range is small, but we can recreate the experiment in a computer simulation of particles obeying Newton's laws, being vibrated, colliding inelastically, etc. Complete mechanical recreation of the experiment. And what we can do is we can measure the displacement field everywhere, and we can ask, so I claim from the theory that I just wrote down that the decay behind is only as 1 over the square root of x, and the decay in front is exponential. So I'll take the measured displacement field, multiply it by square root of x, and ask what it looks like behind and in front. If I multiply it by square root of x, it should be non-decaying behind the rod, and sure enough it is. And it should be decaying in front, and sure enough it is. So now, calculate from a very simple, very general theory. We can actually confirm this by direct measurements on a physical recreation of the experiment in the computer. Okay? What happens if you put 2 rods into a medium? You can work out that as a result of the tendency of rods in our theory to align along the axis of uniaxial strain in the medium, if you put 2 rods, they will approach each other. So you put them side by side here, they'll start walking, and as they walk, they will reorient and capture and bump into each other. So this is very much seen in the experiments also. The experimental setup, by the way, is like this. You've got a mechanical shaker, an industrial shaker. The experimental geometry is such that you've got the gap here is slightly more than the diameter of the rod. You shake it up and down. This rod tilts a little bit and starts walking in the way that I showed you in that movie. And the beads are, again, of a diameter slightly less than this gap. And that's the system. You vibrate it at a couple of 100 hertz by an amplitude of a very small fraction of a millimeter. The rod walks because the 2 ends of the rod are different, and it tends to most likely come down with the fat end down, and it gets impelled along its length. The spheres don't move because they're spheres. A tilted sphere is still the same thing. So this is the mechanics of why the rod self-propels. And in experiments as well as in simulations, as I said, if you put in 2 particles side by side initially, they turn and capture each other. In the fluid phase, if you put them side by side, they turn away from each other because of the flows. And the most fun thing is that if you now put in 2 rods, one of them initially pointing, let us say, this way, and one pointing this way, this guy moves along, and because the disturbances it creates are highly screened in front, this other guy hardly responds to the approach of this one. And this one, on the other hand, can sense the trail left. The one approaching from behind can sense the trail of the one that was in front. And so what happens is, you get a highly non-reciprocal behavior. This guy responds and turns towards that guy. That guy is likely unaware of the approach of this one. So you can have something sneaking up on something in front of it and bumping into it. So you see it, you predict this in the theory, you see it in great detail in the experiment and in the simulation. And so it's an interesting new mechanism. And if you like, it's not actually an attractive interaction. It is much more like what you call, in what some biologists have called elasticotaxis. It's a reorientation in response to spatial variations in the elastic distortion of a medium. And this non-reciprocality is one of the very pretty features of this system. So in the time that's left, I will not go back to the story about defects. I'll tell you about a very different system. Now I'll talk about chiral active matter. You know the object is said to be chiral if it and its mirror image cannot be superposed one on the other, such as these two left and right-handed shells. In 2 dimensions, chirality is the difference between an S and a Z, if you like. And in 2 dimensions, therefore, you have to have an object that's clearly this object can be made to look like a Z by going to the other side. So to define chirality in 2 dimensions, you have to think about looking at a surface and having a distinction between up and down on that surface. Now, supposing you make a physical system out of chiral particles, what are its general properties? One famous example of a chiral system is a cholesteric liquid crystal. It's made of chiral molecules. The optical axis or the physical axis of those molecules turns as you go from one layer to one position to the next in a systematic periodic way. So if you like, it's a system in which the optical axis statically has an orientation wave. There are no true layers, but the macroscopic mechanics of the system at thermal equilibrium is essentially like that of a layered liquid crystal. However, and the chirality doesn't show up in the long wavelength mechanical properties of this system at thermal equilibrium. However, what we show is if you take the system and incorporate 2 features into it. Remember, as I said, it's effectively like a layered system. If you remember that any system with an axis, if it is active, has a stress oriented in the direction of the axis, you can guess that a layered system could have an instability in which it spontaneously undulates the way I showed you. You can get a splay instability for orientable objects. That's one feature. The second feature is that if you have these spontaneous undulations, then it turns out there's a very funny force that comes out of our equations of motion. If you have a local bump, that means you have a local curvature, you have an active force that points along contours of constant curvature, points tangent to contours of constant curvature. So if you have an undulated state like this, then locally, if you've got a bump, you've got a force pointing perpendicular to the direction in which the height grows. So you get an undulated state and you get a state in which you get forcing perpendicular to the undulation. So you end up getting a spontaneous instability to an undulated state together with the circulation. So you get the spontaneous formation of an undulated state with circulating flow, a vortex lattice of flow. So we can create such states. We can actually control them because you can suppress the undulation instability by compressing the system and making the instability go. We have in cholesteric liquid crystals, if you could introduce active stresses, either by releasing microorganisms that stir the system slightly or by other active processes, you can create a system in which you have an on-off switch for a flow vortex lattice. So this is something that you can read about in a paper on the archive currently. So I've tried to tell you about our very latest work. This is work done by my student Swapnil Kole with Gareth Alexander and again Motile. You can see similar interesting effects in 2 dimensions, which I will not point to because I would like to leave time for questions. So I will not go back to the nematic and defects. Instead, I'll close my talk here so that there's some time for discussion and questions. So I hope I've convinced you that the ideas and methods of condensed matter physics can be applied to many aspects of living matter, provided the topic that you're interested in is energy conversion and movement. That is, if you focus on motility and mechanical forces and leave aside the really profound questions of information processing, evolution, and so forth, then you can say quite interesting things about living matter. I told you about our recent, I summarized our old work on it, then I told you about our recent work on how you can actually get stable flux in fluid if the particles move rapidly enough, and you can get a transition from turbulent to non-turbulent, through which you can get a flocking transition. I did not tell you about motile topological defects very much, except at the beginning. I told you about how inanimate particles can chase and capture each other by responding to the elastic strains of an ambient medium. It seems to me that biology should somehow make use of that idea, maybe of motile cells moving through epithelia or moving through the extracellular matrix. And I told you about how to create controllable self-stirring patterns in a certain kind of chiral active matter. And I'll stop with that and take questions. Thank you very much.
So, thank you.
Should I stop?
Professor
Should I stop?
Ramaswamy.
Or should I keep the share on?
Yeah, I think you can stop sharing the screen.
I think you.
Great, yeah. Okay.
Thank you. That was really a wonderful lecture, and I'm sure there will be some questions. Chandrima Shaha, would you be able to?
Yeah, there were some restrictions on the YouTube and I couldn't get the questions. I don't know what kind of.
Okay.
But I can. I have a bit of questions.
Sure.
I'm totally. I mean, I from Professor Sood's lecture got some idea about these active matters, but the microphotograph, was it a scan? I don't recall. The nucleus that
Yeah.
cell. So it is within a confined. It is within a confined active fluid, right? There is fluid inside the nucleus. There is fluid outside the nucleus.
So this is, I mean, these were fibroblasts on some sort of fibronectin pad of fixed shape, and the image was only of the cell nucleus. So the cell interior is also a kind of active complex fluid. I mean, the inside the nucleus also, you can think of it as an active complex fluid.
Yes.
But it's much more viscous and much more hard to deform than the cytoplasm. So what was happening in that, and we actually did fairly detailed theory on the flow field, is that the actin filaments in the surrounding medium were kind of aligned, and they were undergoing the same spontaneous flow and slight distortion instability that our theory predicted for simple linear geometries. So I don't know if I have slides with that here, but basically the idea is this: what was happening there, which we sort of evidenced in great detail by measuring the flow. This was experiments by Shivashankar then at Singapore. The actin filaments in the surrounding medium underwent the instability that we had calculated, as a result of which you got spontaneous flow in the surrounding medium with a characteristic velocity profile that we could calculate and they could measure. That flow, because the nucleus isn't completely anchored, that flow rotated the nucleus. So what you were seeing was the nucleus rotating, and it was rotating because of coherent flow in the medium, in the intracellular medium around the nucleus. So that's what is happening. And both the magnitudes and the velocity profile actually corresponded rather nicely to what was measured.
So during development, that when the tissues are actually growing and they separate out in different cells of different shapes and sizes, and so that's a very interesting area where one can look at the active matter, how it is playing a part in developmental setups. Are you aware of?
Yeah, so actually the group at Max Planck Institute for Complex Systems and MPI for Cell Biology in Dresden are very much using this approach to understand development. So they actually take a hydrodynamic approach to developing tissue itself. There's two sources of active stresses, if you like. First of all, there's actomyosin contractility, but there's also cell division itself. So cell division, every time a cell division event takes place, you get a local uniaxial forcing. That's one kind of activity. And the other is actomyosin contractility and myosin transport and so forth. So also this kind of, so Madan Rao and Thomas Lequi have also been doing theories of basically of in the context of development using this sort of framework, but augmented by many more, somewhat more realistic features that have to be put in. So it's, yeah, you're quite right to point that out, but in fact, there are people doing that kind of thing.
Yeah. So can active matter be used as therapeutics? Like, for example, during malignant growths, can their motility be manipulated to act as therapy?
Well, okay. So if you know Ambarish Ghosh at IISc, he has been so far, I think, trying to use externally actuated submicron particles to deliver either, you could imagine either delivering specific compounds to a location, or you could imagine delivering particles with a certain property there so that maybe they can take up more heat. So you could maybe ablate tissue in some place where something is bad. So these kinds of things are being tried. I don't know if fully autonomous self-propelling objects have been put to work in this way, but that is certainly one of the reasons why people are interested in it. And on the Indian scene, Ambarish is the person to talk to about these kinds of applications. So short answer is yes, it's, yeah.
Yes, it can be. Yeah, this is very interesting. I mean, this is a field where, how young is the field?
So it depends. I'll tell you. So I think the name, I did a little bit of archaeology on my, on the net and on my Gmail folders. And the earliest reference to the term active matter that I was able to find was in an obscure paper by Aditi Simha and me from 2006. Not in an unusual setting in solid-state communications, where for some reason they decided there should be a special issue on soft matter. So we wrote something up, and that's the first place I am able to find the term. Several people have asked me who first used it. As far as I know, we were the first. And but the subject of looking at self-propelled particles and their organization in the physics context goes back to Vicek 1995 and Toner and 2. So they looked at flocks. And the difference, if you like, between those approaches and ours is that mechanics, the actual mechanical consequences of movement, forces, stresses, and so forth, was absent. Theirs was an agent-based or rule-based model. And the rule wasn't Newton's laws, it was something looks at its neighbors and aligns. So in that sense, it was different.
Okay, so thank you very much for this lecture, which I mean, many aspects, I think the active matter is going to be very important in future therapeutics also.
That would be great.
I think we are far away from what I'm saying, but I'm sure it has a tremendous potential in using body's own mechanisms to do therapy, and also inherent active matter, manipulating inherent active matter. So
I had a question.
Sure.
yes.
Sriram?
Yeah.
That R parameter that you defined for the systems with inertia, it looks a lot like Reynolds number to me.
Good question.
Going exactly the opposite way of the Reynolds number. That low Reynolds chaos and high Reynolds number, no chaos.
Yeah, so it's a little tricky to know whether it is exactly a Reynolds number or not. I'll tell you why. Maybe I'll just share the slide again.
Sure.
Yeah.
Okay, let's see. Just a second. Okay, so can you see the slide?
Yeah.
Okay, let me just skip ahead to 1 sec. So here, so it's the you can view it either as a ratio of 2 squared speeds or as a ratio of 2 stresses. Now the question is, how do you estimate the active stress associated with the swimmer? If the swimmer is purely, so this is a totally technical answer, but it needs, unfortunately, it needs a totally technical answer. I can't help it. If the, in principle, within a phenomenological coarse-grained theory, the self-propelling speed and the force dipole strength are independent parameters. You can imagine a swimmer with no force dipole, a pure force quadrupole. Imagine a torus which creeps along its, kind of longitudinal direction. You see what I mean?
Yeah.
That is a pure quadrupole swimmer. So you can imagine, and also it's important to note that you can imagine, so basically you can imagine a swimmer in which the force strength is as small as you like, and the primary swimming mechanism comes from some higher order multipole. So that's one technical way in which you can have independent control over the self-propelling speed and the force dipole strength. The other is that there's the question of how do you estimate the intrinsic active stress associated with the swimmer. If it is a purely viscous swimmer, then you will say this active stress is basically given by the viscous stress on the scale of the particle, and then this will become exactly the Reynolds number on the scale of the particle. If the swimmer has a moderate amount of inertia, then it might be that this active stress is really primarily inertial, and then this thing becomes of order 1. So there's some slack in exactly what the value of these guys are. But probably the best way to approach the problem is experimentally to find a swimmer whose Reynolds number at the particle scale, rather than being minute like for a bacterium, is kind of order 1. So there is a group, there's Daphne Klotza at University of North Carolina, I think, who now is trying to create swimmers of that type, either artificially or by looking at organisms, so that one can start to push active hydrodynamics with experiments which venture into the regime where this Reynolds-like parameter is not 10 to the minus 4, but order 1. So I think that's the very detailed and kind of nerdy answer to your question.
Great. Thanks a lot. So thank you very much. And I think, unfortunately, we were not able to get hold of the questions on the YouTube channel. So we'll end the session today.
Sure.
The lecture will, of course, be available online.
Yes.
Lecture also.
Sure.
And
Yeah.
hopefully some of the people who have questions can contact you directly.
Yeah, sure. They can certainly send me mail. Thanks very much. I really enjoyed it.
Thank you very much. Thank you.
Wonderful lecture.
Bye.
Very insightful and nice lecture. Thanks a lot.
Thank you very much. Very kind of you. Bye.
Bye-bye.
I'll disconnect. Yeah.
Bye. Bye.